Our next speaker is Dr. Jennifer Rakeman Cagno, who is the Senior Director for Medical and Scientific Affairs under Public Health Programs at Cepheid. And in her role, she is focused on advancing diagnostics for infectious diseases of public health importance and pandemic preparedness. So I'm going to turn it over to her to share Cepheid's perspective on the expert HCV journey and what it was like going through ITAP.
Great. Thank you so much, Nate. And thank you so much for having us as part of this amazing webinar today. So we can go to the next slide. And as I talk about and thought about the Cepheid journey here, I really wanted to start with Cepheid's mission, which is delivering a better way to improve patient outcomes by enabling access to PCR testing everywhere. And the reason I wanted to reference this is because this really does embody the goals of this project in really ensuring that we could get this test that, as others have mentioned, had been available outside the US, getting it available in the US to patients who need it where they are. And so if we go to the next slide, I just want to talk a little bit about the history and the background as we came into this project. So as Dr. Collins alluded to, the Cepheid had an HCV viral load fingerstick-based test available outside the US since about 2018 and had been commercialized across Europe, Africa, and Australia. And so in that sort of 5-year period before we thought about this project, we had learned a whole lot. And the test had had a very big impact in these other locations outside the US. And what we learned was that if a site had access to the instrument and the test, was able to prescribe treatment, and had drug available, that really enabled this test to treat paradigm where patients could come in or be met where they are, get a test, get a diagnosis, and get linked to care and linked to treatment within a visit. And we sort of obviated those issues that resulted in loss to follow-up. We also learned that phlebotomy is challenging. Eric alluded to this as well, not only because it's sometimes challenging in patients, but also because it's challenging to do in very decentralized settings outside of a clinical site. So capillary blood is really helpful because a fingerstick collection can be done by a community health worker or a non-phlebotomy trained person and can be done in these decentralized settings. So if we go to the next slide, this just shows one of my favorite projects outside the US, which is the Combi Clinic in Australia. So in this case, the team works out of a 1975 Volkswagen bus. They have GeneXpert instruments on the bus, and they take it out to where patients who need HCV testing are. They meet them where they are. They do the testing on the bus. They get the result back to the patient and get the patient linked to treatment. So they walk away from that bus, if positive, with their first days of medication so that they can move towards a cure. So we can go to the next slide. So kind of where we started and the level of collaboration that happened in this project was just absolutely incredible. So as mentioned in early 2023, we had a conversation with the Federal Task Force, Dr. Collins and team from the White House, about this interest to bring this fingerstick test that was available outside the US to the US to really focus on elimination programs in the US. Throughout the project, there was an incredible level of collaboration across the federal government and others really to make this happen. So Cepheid's job was to sort of make the test, make the tool, but we needed all of this other collaboration to be able to ensure that the tool could be used. So the ITAP team here is in a big red box because they were just central and really important to this interaction that happened between Cepheid and the FDA and our work with NIH. We worked with CMS to ensure the test would be eligible for coverage and reimbursement. We worked with CDC and AASLD, and you'll hear from Nate and Andrew next around the guidelines that drive how clinicians and providers can use the test. And of course, we worked with our congressional partners around the elimination plan legislation. So we can go to the next slide, which is a timeline of kind of how things went. And this is not to scale and very, very abbreviated, but kind of on the left side, you sort of see where we started with this information and experience outside the US. A number of things happened early in the early 2020s. So there was the strategic plan that was released for hepatitis elimination. Importantly, the FDA downclassified HCV RNA tests, which allowed the opening of the regulatory pathway, also allowed for the test to potentially be CLIA waived and available in point of care settings. We had those early conversations with Dr. Collins and the White House team. And then in January 24, we launched the very beginning of the clinical trial for this test in the US. And as Eric said, at the end of June, we had an approval and authorization from the FDA, and we launched the product in July. We can click one more time. Really, that mindset and the Cepheid team in that period was we just had to shrink the timelines. Things that usually took a month or months became a week. Weeks became a day became a few hours. And as Eric said, we went through this with the mindset of there is no July. This was happening by the end of June. It took a ton of work. A lot of the Cepheid village was involved in this, many nights and weekends. And again, that collaboration with all of these teams across these partnerships was just really important for this project to move forward and for this to happen in the time frame that it did. So if we go to the next slide, this just shows with the workflow of the test. As Eric mentioned, we had this challenge with the collection device, and we had to essentially validate a collection device that was not approved in the US for use for molecular diagnostic testing for infectious disease, the microtainer. So the blood is collected, it's pipetted into the cartridge. This is a cartridge-based test. The cartridge goes into the instrument, and then the instrument runs the assay and provides a result. So it's a very simple workflow, but we did have to work collaboratively with all of these partners throughout this process to ensure that we had the right materials, that the instructions for use and the quick reference guides were appropriate for people to be able to run the test successfully in these decentralized settings. If we go to the next slide. So the top of the slide shows the sort of more traditional timeline for HCV diagnosis that Dr. Collins referred to. It could take days to weeks and multiple visits for a patient to get a diagnosis of HCV and then get linked to care and treatment. And at any one of those steps, any one of those arrows, a patient could sort of fall out and be lost to follow-up. So there's lots of opportunity for patients to be lost to follow-up. And what we have now in the red circle in the bottom of the slide is the ability to do a diagnostic test at the point of care where a patient can walk out of that visit or walk away from that site, knowing their HCV infection status, whether or not they have an active infection. And if they are infected, they can walk away either with a linkage directly to care or with medication in hand, so that we can get more people tested, more people treated, and more people cured and move towards that elimination goal in the US. If we go to the next slide, my thank you slide, there are many, many others that are not mentioned in this alphabet soup on this slide. This is not meant to be comprehensive at all. We had a huge Cepheid village, as I mentioned, working on this that worked really, really hard. We worked with all of these people. I also want to call out the ITAP team. We spent many hours a day working closely with them. They worked really hard to get us answers, to escalate things as we needed. It was just an incredible, incredible collaborative set of work to make this happen. So I will end there and pass it back to you, Nate. Thank you.